Yeah, guys, let me just clarify for y'all. You guys can use the chat, or you can use your microphone to ask Mr. Sharp any questions you guys have. Either one works. Whenever you're ready to start speaking, just go and start speaking. Oh, there. All right, Mr. Sharp, you have a question in the chat. Just go and look over there. Hey, Mr. Sharp, are you there?
Somehow I got muted about that. Sorry, I was talking and apparently I got muted. Okay, so I'll go back and say it again. So the question was, how do you tell the difference between polar and non-polar R groups? And the way you, what you should be looking for, your clue is you should be looking for an electronegative atom like oxygen or nitrogen or sulfur in those R groups. So you want to make sure you're looking for those. If you see an R group that has a hydroxyl group or an amino group like an NH or something like that, then you can assume that that is a polar side chain. The ones that are non-polar are going to be pretty much all hydrocarbon. So nothing but carbons and hydrogens, especially when they're in those ring structures. Those ring structures make them very stable. And so then those will be more non-polar. Why is there a space for the non-competitive enzyme in the first place? Are you talking about maybe like the non-competitive, the allosteric inhibitor on an enzyme? Okay, so, well, nature is nature. It's not like the allosteric site for where a non-competitive inhibitor can bind to an enzyme was placed there on purpose. Oftentimes, plants and animals and other living systems, they develop these things as a defense mechanism, right? So a plant has poisons and toxins that it creates chemical defenses that can interfere with or annoy herbivores, right? So if they don't want to get eaten by deer or little critters anymore, over time they might develop a chemical defense that somehow binds to an enzyme in a deer system or another mammalian system that annoys them or causes them pain. And then if that stops working, then, if it stops a pathway in their body, then that would be good for the plant. Mostly these are happy little accidents for the plants that come up with these chemical defenses. So there's all kinds of different ways. Now, remember, not all inhibition is bad. It's not all poisons and toxins. We actually regulate some of our own enzymatic pathways, our own metabolic pathways, purposely. So the end product in those feedback inhibition systems, the end product will oftentimes inhibit one of the enzymes early on, and oftentimes it does that at an allosteric site, so somewhere other than the active site. So it could be just to regulate chemical activity inside the cell as well.
Share screen. Talk. What? What? Wait. No. Why can't we hear? Wait.
We can hear you guys.
Oh. Oh. Okay. There we go. I really hope that.
Go ahead and ask away. Don't be shy, people. Really, Drew? All of cellular respiration? Okay. So remember, the key things to remember from cellular respiration are the inputs and the outputs. But don't worry, I'm not going to ask you to try and recite every single step, or how many NADH or how many FADH2 come out of this Krebs or glycolysis. I'm not going to ask you to name all the members of the electron transport chain and oxidative phosphorylation. Keep an eye on the big picture, right? The whole point of cellular respiration is that we need to make sure that we're breaking down food and making ATP. So you keep that in mind, right? The whole point there is to make ATP so that we can do cellular work. Then you'll be okay. Now, as far as what you need to know from it, it is an enzyme-driven activity, right? It's a process. It takes a whole lot of little individual chemical reactions and kind of puts them all together in one big thing. But you need enzymes all through there, right? So enzymes need coenzymes to help them work, and they have to be in the right environment. So we do all kinds of things to regulate cellular respiration. But big idea is that we are ripping electrons off of food molecules. We're going to oxidize food, removing those electrons. We're going to place them onto electron carriers like NADH and FADH2, and then we're going to deposit them into the electron transport chain. The electron transport chain is embedded in that inner mitochondrial membrane, and that's going to build the electrochemical gradient, that proton gradient. And we're going to store up that potential energy by separating those two environments and having the intermembrane space be full of hydrogen ions, and the mitochondrial matrix have a low concentration of hydrogen ions. And then we give them a channel to get down through there, right? So we put ATP synthase in that inner mitochondrial membrane, and then as the hydrogen ions flow downhill, they will produce ATP. Someone else asked if we need to know the difference between enantiomers and isomers. No, there's nothing on here about that. It's not a bad idea to remember that enantiomers are stereoisomers, right? They're the ones that are mirror images. So you just can't, they would look alike if you put them in a mirror, but you can never superimpose them on themselves. But no, there are no isomer questions on this particular file. Oh, okay. So 4 levels of protein structure, that's a great question. So remember that all proteins, right, they start off as amino acids, right? So individual amino acids are hooked together via peptide bonds, which are strong covalent bonds. So that's our first level of protein structure. It's just that linear chain of amino acids in the amino acid sequence made at the ribosome. And it's just held together by covalent bonds, those peptide bonds. The second level of structure in proteins is the association of beta-pleated sheets and alpha helices, and those form and are held together by hydrogen bonds that are hooked up on the backbone of that amino acid chain. So by the backbone, I mean, when you look at that common structure that all amino acids have, with a carboxyl on one side of your central carbon and an amino group on the other side of your central carbon, and then the hydrogen coming off the bottom, that is the backbone. So interaction between the carboxyl groups and the amino groups all along the overall length of the amino acid sequence, they, depending on the order they're in and the type of environment they form up in, they will make those helices, those helix shapes, and those beta-pleated sheets, and that's your secondary level of structure. So the first level of structure is just amino acids linked together at the carboxyl and amino groups by covalent bonds. The second level of structure is held together by hydrogen bonding along the backbone. The tertiary or third level, that's when we start using those R groups. So that's where the polarity of the R groups, like knowing that the polar versus nonpolar and the acidic and basic R groups, that's where it becomes important. Because once they start interacting with each other, that's how we form that tertiary level, that third level of protein structure. And then when we start putting those three-dimensional, those 3D folded subunits together, we get to our final quaternary or fourth level of structure. So the forces that hold together the tertiary, they could be weak ionic, they could be hydrogen bonds between polar groups, they could be that disulfide bridge between two cysteines. We get that sulfur-sulfur covalent bond, which is very strong. And then they could also have those the van der Waals interactions holding together when you have the hydrophobic groups that are all grouped together. Then you get those van der Waals forces. So those are pretty much, the majority of the forces that are going to hold together the tertiary and then also that quaternary level of structure. Ria asks, is it just the levels of cyclin control? They control all the checkpoint gates in cell division, or is there something else that helps control the checkpoint gates as well? So, we really just kind of scratch the surface when it comes to checkpoints, because checkpoints can be a lot of different things. It can be an accumulation of different types of chemical signals inside of the cell. And so for us, let's just focus on just saying they have achieved whatever, they've accumulated enough of whatever it is to pass the checkpoints. But cyclin is absolutely involved in both the G1 and the G2 checkpoints. They combine with those CDKs, the cyclin-dependent kinases. There's just different types of cyclin. There's G1 cyclin, and there is mitotic cyclin, and it is mostly the same thing, but they are slightly different, and they bind to those CDKs in a little bit different way. All right, Emma, Maria, and Ivy, they asked, what is dehydration synthesis? So remember, that goes back to our Unit 1. When we are linking together monomers, we use that process of dehydration synthesis, where an enzyme will take a water molecule out of, basically it takes an OH off of one monomer and a hydrogen off of another monomer and puts them together. So that's the synthesis part. The dehydration part is we take that OH and the H and we make a water molecule out of it. So we're removing a water molecule from the two monomers as we hook them together. So think of dehydration reactions are going to build things up, where hydrolysis is going to break things down. Okay, so dehydration synthesis will build up polymers, and hydrolysis will break polymers down. And I don't know if you heard it, but Chester the kitty cat is walking around and trying to talk to you guys. Allison asking about the FRQ. It depends on how much of your structure and function you have on that chart, right? So you definitely want to look at structure and function beyond just, this is the structure of this particular organelle and here's what it does, right? I want you to be able to link that structure with that functionality. So if you're talking about something that's made up of proteins, like an enzyme, or if you're talking about something that has, you know, if your question was about like a cell membranes are made up of multiple different types of macromolecules. So understanding, you know, in a membrane, the structure is mostly phospholipids. And the functionality of that is because phospholipids are hydrophobic, they have that hydrophobic region in there. When we have a phospholipid bilayer, that will maintain a separation, right? It is a boundary between two different environments. And that way we can have a difference on the inside of the cell and the outside of the cell. We can maintain different sort of solute concentrations. We can have different sorts of, we can build that electrochemical gradient. We can have more or less water, whatever it is. But make sure that you're not just memorizing what the structure of something, you know, how something looks and then what it does, but making sure you understand how those two things are related to each other. All right, Emma Maria and Ivy ask, do we have to know the specific inputs and outputs for glycolysis and the citric acid cycle? No, don't get caught up in memorizing like individual details like number of NADH or FADH2. You know, I do want you to know that, you know, coming out of glycolysis, we're making pyruvate. We're converting glucose into pyruvate. And then pyruvate has to be converted into acetyl-CoA before we can start the Krebs cycle. But I really want you guys to focus more on understanding that it is a process. And the whole point of this is to take those electrons off of our larger molecules in a series of reactions and then just putting them onto those electron shuttles, the NAH and the FADH2. Getting them over to the electron transport chain at oxidative phosphorylation so we can build up that electrochemical gradient. Yeah, so Drew, you're right. In terms of genetics, just simple Mendelian inheritance patterns, right? So dominant, recessive, the same stuff that we had on the quiz last week. I'm not going to go beyond anything after the quiz. So just knowing basically monohybrid crosses with, you know, only looking at one character. And if there are two traits, then we would have two alleles, and that's all we're going to have, right? No multiple alleles or complex inheritance patterns. It's all going to be pretty much straightforward right now. All right, so Taylor says, can you explain what concentration gradients help to do? Okay, so, well, if you have a concentration gradient, that simply means that we have more of a solute on one side of a barrier, like a selectively permeable barrier, like a cell membrane or mitochondrial membrane. So if we have more of it on one side than on the other side, then there is potential energy there, right? It's the potential energy stored up in that position, because what do we always want, or what do we know that molecules always want to do, right? They always want to reach equilibrium. So if we can give them a pathway, they will flow downhill, right? So if they're charged particles, and that's, you know, it's got two forms of potential, right? It's got the electric potential, and it's also got the chemical potential of the concentration gradient. So when we are storing up like an ion concentration on the inside and outside of nerve cells, that's going to give us the potential for an electrical impulse that can send a message down a nerve cell. We're more concerned right now with this in this semester looking at the concentration gradients stored up across the inner mitochondrial membrane, and that is the hydrogen ions, right? And so when we pump those hydrogen ions into that intermembrane space, we now have a chemical gradient. And if we let those hydrogen ions have a pathway back into the matrix, then we can harness that kinetic energy of them moving, their kinetic energy of their motion, and we can use that to let ATP synthase make ATP. All right, Akshay says, can you go over the signal transduction pathways we need to know and how they work? All right, so as far as the signal transduction pathways, I mean, I'm not going to ask you to like specify one. Like, you're not going to have to like to do the epinephrine pathway from start to finish or anything like that. It's mostly just understanding that we have 3 phases to signal transduction pathways, right? There's the cell receptor, there's the signal reception phase, there's the signal transduction phase, and then there's the cellular response. And on those receptions, we have, you know, different types of receptors. We have the ones that are embedded in the cell membrane, showing up on the outside of the cell, like the G protein coupled receptors and the RTKs and the ligand gated ion channels. And then we have the ones that are inside the cell, those intracellular receptors. And they're going to be looking for a hydrophobic or nonpolar messenger molecule. But the ones we spent the most time talking about were the GPCRs and the RTKs, right? And those are cell surface membrane receptors or cell surface receptors. And so their ligands are going to bind to them on the outside of the cell. So that's really what I'd rather be focused on, because there's a clear delineation there. There's a clear like line of demarcation between the reception phase and the transduction phase. The reception is just getting that message from outside of the cell. You know, that ligand, that first messenger, if it's polar, it's not going to be able to get through that cell membrane. So that cell receptor has to be on the message receptor has to be on the surface of the cell. And then once we move inside and we start changing the shape of those relay molecules or those second messengers, that's when we have entered into the second phase, right? The signal transduction. Because the signal itself, once we move inside, the signal itself is literally the molecules that are being changed. And when their shape is being changed, that is the signal changing. So we have those protein kinases and that phosphorylation cascade. Every time we change the shape of a protein by adding a phosphate group to it, that is the signal. So the signal keeps getting passed along with the change in shape of those relay molecules. And then you got your cellular response is almost always going to be either gene expression of some sort or activation of an enzyme inside of the cell. All right, let's go back up here. Allison asks what the van der Waals interactions are. So remember, even in nonpolar bonds, or pretty much in every object, right? Electrons are moving, even if they're not being shared unequally. If they're being shared equally, they're still moving around inside of their electron shell. So there are very, very small, brief moments and very like localized areas of those partial charges, just simply because the electrons are always moving. So we always have some very, very, very small, very weak partial negative and partial positive areas of adjacent molecules. And so things are really, really closely together, all those little tiny van der Waals forces, they can hold things together. So when we have hydrophobic interactions, that just simply means that the areas on those proteins, like the R groups, if they're hydrophobic, 77% of the cell is water, right? Every cellular environment is very, very watery. Your blood is watery. You're basically a big bag of water. So those hydrophobic side groups and those hydrophobic areas of proteins, they have to get away from water. So they will basically be forced into the same area. So they're always forced very closely together. And because they're so close together, that's where we get those van der Waals interactions. Remember, a lot of really small interactions, they can be strong enough to hold things together, hold things up. You know, van der Waals forces are how insects and bugs and stuff can crawl up walls. It's how geckos can crawl up walls. They have all kinds of little indentations in the pads, and they have little hairs on the pads of their toes. And all those little slight imperfections, they meet with the wall, and they use van der Waals interactions, the van der Waals forces, to hold them on the wall. Okay, Cecilia says, can you go over what conditions help enzymes work best for catalytic reactions? Well, that's going to depend on the enzyme, right? So remember that the enzymes that work in your stomach, so gastric enzymes, they work best at a very, very low pH, in a very acidic environment. So that's why we crank those stomach acids into our stomach. So the stomach acids themselves don't break down food. They just set up the proper environment inside your stomach for those peptidases and other enzymes that work in your stomach to go to town on the food that you just ate. So really, it's just, if I said, how do you set up the right conditions for enzymes, and you would say it has to be the proper temperature or the proper pH or the most effective temperature, most effective pH, because it can change. The enzymes that work in your small intestine, they work best at a slightly basic environment. So your intestinal pH is around 8 or so, whereas your stomach is down around like 2. So it all depends on the enzyme itself. Now, the vast majority of the enzymes inside of your cells, they work best at a pH of 7 because most of your cell is water. So they are going to work at a pH of 7. But the hydrolytic enzymes inside of your lysosomes are kind of like the stomach enzymes. They work well in a low pH environment. So we actually have proton pumps on the surface of our lysosomes, and they will pump hydrogen ions in there to try and lower the pH. And by lowering the pH, they set up the right environment for those enzymes. So making sure that you have the right conditions for enzymes is mostly about ensuring the correct pH and the correct temperature. If it's the wrong pH and the wrong temperature, then you will start to denature those proteins by breaking apart some of those weak interactions that hold them together, especially those hydrogen bonds. And if we change the shape of something, we change its functionality, right? Because structure and function go together. So when things get denatured, they start to break apart. All right, so Praneeth is asking about Anchorage dependency, right? So Anchorage dependency, that was in our mitosis unit, right? Or mitosis chapter of unit 4. So Anchorage dependency is basically just saying that mammalian cells especially, they have to have a substrate to grow on. Like they don't just grow when there's nothing there, right? So if you put human stem cells on a surface that didn't have the ability that they could not stick to, or they couldn't get nutrients from, then they wouldn't be able to grow there. So anchorage dependency simply means there has to be something for those cells to anchor onto, so they will grow, and they will grow until they cover it, right? And then once they're covered and there's no more area to grow into, they'll stop growing. But basically, it just means you have to have like a substrate. You have to have something for them to grow on. Ria says, what's the difference between lysosomes and peroxisomes? Peroxisomes are they are a bit more specialized. Lysosomes can bind with, or they can fuse with like food vacuoles. They can fuse with vacuoles that are bringing in foreign invaders, like in white blood cells. They can fuse with vacuoles for autophagy, when they are recycling old, worn-out cell parts. Like if you have a mitochondria inside of a cell that's no longer really functioning all that well, then it will fuse with a lysosome. And lysosomes have those hydrolytic enzymes in them that can break down basically almost any covalent bond. So they'll break down all the macromolecules that we find inside of cells, whether it's our friends, right, like our own organelles, or whether it's a foreign invader. Like if you got a bacteria in you, or you got a virus inside of you, or you got a mold or a yeast inside of you. Any of those things, they're all going to have, carbon-based macromolecules, and hydrolytic enzymes found in the lysosome can break those things down. Peroxisomes are a bit more specialized, right? So they're going to go after, they help to break down more specific metabolic byproducts. And they're special in that they're the ones that will create hydrogen peroxide as they break things down. And so then you also have to have catalase in there to quickly convert that hydrogen peroxide into oxygen and water. All right, you guys are getting some good questions in here. Can I explain the importance of surface area to volume ratio for cells? Sure. Remember, as an object gets bigger, right, as a three-dimensional object grows, the volume is going to grow faster than the surface area will. So pretty soon, it's going to take more time to get things from the outside of it all the way down to the middle, or to get things from the middle all the way to the outside. So as the surface area to volume ratio decreases, meaning that the whole entire object is getting bigger and bigger and bigger, that means that we're going to have a more difficult time getting nutrients all the way in and getting enough nutrients all the way in and getting the wastes out. So we have to maintain the proper surface area to volume ratio by having enough area close enough to basically all the inside parts to be able to make sure we can get food and nucleic acids, or nucleotides, I should say, to build nucleic acids, and amino acids to make proteins, and get enough of that stuff into the cell and spread it around all inside there, and get wastes like carbon dioxide and other metabolic waste products out of the cell. So it's basically to make sure that we have a proper exchange of nutrients and wastes. Importance of the sodium-potassium pump. Again, sodium-potassium pump is the main pump in mammals and mammalian nervous systems. It helps to set up the proper balance of potassium ions and sodium ions on either side of cell membranes in the nervous system, helping us send those action potentials and send those nerve impulses all over our body. That's the main importance of the sodium-potassium pump. What I want you to know about the sodium-potassium pump, right, is that it is a pump, so it is active transport. It requires the input of energy from ATP molecules, and it's, from a structural standpoint, it is a transmembrane protein. So it's going to have portions of it that are hydrophilic and portions of it that are hydrophobic. It also allows items to pass through the cell membrane. So obviously that the channel inside there, which changes shape, has to be full of hydrophilic sections or R groups, amino acids with hydrophilic R groups. It also has very specific binding sites. So going back to structure and function, right? It's got 3 binding sites for those sodium ions to leave the cell, and it has 2 binding sites to bring potassium ions into the cell. So that's a good one to talk about structure and function, because it has the exact right structure to fit sodium ion and the exact right structure to fit a potassium ion when it's in that position. C4 in can plants, nothing really. I mean, just know that all plants are going to have evolutionary adaptations to help them deal with varying conditions. So some plants have adaptations to deal with drought, to deal with dry conditions, to deal with hot conditions. And that's the C4 in can plants. But there's no in-depth details or anything you need to know about C4 in can. Justin says, other than temperature and pH, what affects enzyme activity? Something else that would affect reaction rates would be the concentration of the substrate molecules, right? So how many reactants are there? Obviously, the more reactants there are, the more likely we are to have an interaction between the enzymes and the reactants, right? Which become substrate molecules, and then we can create more of the product. So the more substrate there is, the more enzyme there is, that's going to drive up enzyme activity as well. But yeah, you're right. As far as environmental keys and environmental factors, it is temperature and pH, but also how much of the concentration, or what's the concentration of the substrate and then the enzyme. And then also, if there is a competitive inhibitor, what is the concentration of substrate compared to the competitive inhibitor, right? So if it's 50-50, right? If there's the same amount of competitive inhibitor as substrate molecule, then that's going to significantly affect the reaction rate, because then you're like, basically you'll have half the reaction rate because those substrate molecules are competing with the competitive inhibitor for the enzyme's active site. Allison asked, do we need to know about specific heat and evaporative cooling? Just as far as, they're super important. Well, all living things utilize water as a solvent, right? It is the universal solvent of life inside of both eukaryotic and prokaryotic cells. So why is it so important? Number one, for specific heat, it allows a whole lot of chemical reactions to occur because chemical reactions are never going to be perfectly efficient, right? We're always going to give away some heat, right? We'll lose some thermal energy during those chemical reactions. So the water inside of cells can absorb the thermal energy from those chemical reactions without heating up. So we don't increase our temperature when we absorb excess energy from not too much anyway. We don't heat up too much when we absorb that excess energy from chemical reactions. And then evaporative cooling is when we do start to get too hot because we're doing too much metabolic activity. If we're endothermic, like humans and other mammals, or if it's really, really hot outside, like animals and plants that are always out in the open and don't have access to shade or don't have access to air conditioning, then most living things have a system set up for evaporative cooling, and that has to do with water's high heat of vaporization. And so when water vaporizes, when it goes from being a liquid on your skin to being a vapor in the air, it's going to take away some of that heat, right? So it's going to absorb heat energy as it vaporizes, and that will cool down the surface that it's on. So it cools down our skin, it cools down the tongue of a dog when they're panting. It cools down the ears of those long-eared hairs, right? Because they have evaporative cooling going on in those ears. So anytime you can cool things down by evaporation, that will cool down the surface temperature, right? So especially on your skin. Should you know the functional groups in depth? No, there's nothing on there where you're going to have to like name a functional group. There's nothing on there where you're going to have to, there's like maybe a couple of pictures where you might have to recognize some structures, but not a lot. Don't spend any time or don't spend much time at all looking at functional groups. Just remember, if there's OHs or NH2s, if there's anything with an electronegative atom on there, that's going to be a polar side group or a polar functional group. And polar groups can typically form those hydrogen bonds with other polar groups, and they can also be dissolved in water. So that's what you want to be on the lookout for. But don't spend any time looking at that chart or going through them all. All right, you guys were hitting me with a lot of questions there for a while. How can you tell if a structure is polar based off the picture? Again, like I said, you're looking for OH groups. You're looking for, if you're looking at chemical structure, you're definitely looking for, OH groups or nitrogens. Anytime you see an oxygen or a nitrogen attached to hydrogens, then those oxygens and nitrogens are going to be holding on to the electrons with a bit more strength than the hydrogens are. So you'll have those partial positives and partial negatives, and that's where you get the polar nature of those functional groups. But if it was like, if you didn't see the actual chemical structure with the individual atoms and the way they're held together, then you wouldn't be able to tell if it's polar or not. But that's why we have charts, right? So all carbohydrates, all proteins, and all nucleic acids, they are all polar structures. Lipids are all nonpolar. All right, Cecilia, should you know anything about the endosymbiotic theory? No, Chester is saying hi to you guys. Just that mitochondria and chloroplasts were once free-living prokaryotes that were taken up into large, simple eukaryotic cells. And it was one of those cases where it ended up being a better fit for everybody involved. So they stayed in there. They gave an advantage to those eukaryotic cells because they could produce more ATP and produce their own organic sugars and break down those sugars to make ATP. And they didn't have to live on their own and fight and get attacked by things. So they had, I don't have the camera. I'm not going to turn the camera on. You guys want to see Chester? Hold on. Oh, he's not really happy about it right now. Let's see. Chester, look at him. Look at the people. Wave to your people. He's like, let me out of here. All right, I'll put him down now. Okay, I'm going to get rid of the video now too. I hope you enjoyed that. Back to, so as far as the endosymbiotic theory, like I said, chloroplasts and mitochondria were once free-floating, free-living prokaryotes, and now they're part of eukaryotic cells. All right, Praneeth, you've asked a lot of questions. That's good, man. Can you explain the importance of kinetic cores? Absolutely. So kinetic cores are essential in making sure that mitosis works properly, right? They are the area, so the kinetic cores are the large protein structures where the spindle fibers, those microtubules, where they hook up to the centromeres on chromosomes. So it absolutely is essential for all the kinetic cores to be lined up properly, to have the right amount of tension on them before we start the process of separating the chromatids in anaphase. And so they are all talking with each other, right? And there are signals being sent out to stop until everything is lined up properly and there's enough tension on all the kinetic cores. And then as soon as they all get that go signal, that's where the kinetic cores will start the process of, grabbing the microtubule highways, pulling a little bit, and then depolymerizing and breaking them down. So they allow those chromatids to walk all the way along those microtubule highways down to the poles of the cells as we move towards the end of anaphase and then into telophase. All right, RTKs, the receptor tyrosine kinases, and their importance. They're a class of cell surface membrane receptors, or cell membrane surface receptors. They can dimerize. They are often the attachment point or the receptors for growth hormones. So anytime we're talking about growth hormones being the messenger, the protein being sent out to say, let's go into the next phase of the cell cycle, or let's increase cell cycle activity, then that would be the job, or the receptors would be RTKs. From their structure, they had those three. If you remember when we did the activity outside, the RTKs, each one of them had 3 tyrosines on it. And so when each of those 3 tyrosines gets phosphorylated, it can then in turn activate multiple relay molecules. So you can really start a big signal with RTKs. Obviously, with the cell cycle, there's a lot of things that have to get done. There's a lot of different activities inside the cell to activate a whole lot of transcription factors and get things lined up in the proper places, and basically let everyone know that we're about to do a major change inside the cell. So that's why RTKs are important there, because they can start a lot of different pathways from one external signal. The purpose of the bends and kinks on the fatty acid tails and phospholipids, Well, it keeps them from packing too tightly together, right? So when you have those kinks because of double bonds inside those fatty acid tail sequences, that means that they will maintain fluidity. And as we know, the cell membrane has to maintain proper amounts of fluidity. We don't want it getting stuck or packed together. That's why animal fats are solid at room temperature, because they are made of those saturated fatty acid tails. And at room temperature, they get so packed closely together that they solidify, right? And that's why we see like white fat on pork and beef and chicken. But you don't see that on, from plants, right? Their fatty acid tails and their lipids are oils. They're liquid at room temperature because they are unsaturated, right? They have those double bonds in there, and then therefore they can flow past each other. They don't get packed too tightly together. The main difference between cyclic and linear electron flow. So, just think of it kind of in the name, right? So linear electron flow, and this is in the light-dependent reactions of photosynthesis. So the linear electron flow is when electrons flow from a photosystem into the electron transport chain, hop into the next photosystem, and then from there go to the next electron transport chain, and end up at NADP plus reductase, making NADPH. So on the way, they made some ATP, and then they hopped into the second round of electron transport chain and ended up making NADPH. That's linear, it goes straight through. It makes both ATP and NADPH. But remember, in the Calvin cycle, we need a little bit more ATP than NADPH. So every once in a while, the ferrodoxin, which is an electron shuttle, and you don't need to know that name, it will basically shuttle backwards from photosystem 1, goes back over to the cytochrome complex and drops off its electron back over there, allowing for that first electron transport chain to run some more. And remember, the purpose of the first electron transport chain in the light-dependent reactions was to create that hydrogen ion concentration gradient, allowing for us to run hydrogen ions through ATP synthase to make more ATP. So cyclic electron flow is just going to make ATP, ATP, ATP. Linear will make ATP and NADPH. There will probably not be anything about the salmon reproduction as a bonus question, but I appreciate the fact that we enjoy the educational videos that I enjoy showing in class. How does the cell cycle reset for the next signal? Basically, it is the breakdown of those cyclins, right? So remember how cyclin production goes up and then comes down, and goes up and then comes down. Whereas the CDKs, those cyclin-dependent kinases, they stay flat, right? Their concentration stays flat all the time. And so together, when cyclin reaches a certain concentration level, then the cyclin and the CDKs will bind together and make those MPFs, the maturation promoting factors. So when you have both cyclin and CDK, they'll make maturation promotion factors. Those maturation promotion factors are basically transcription factors. They're going to go into the nucleus and they're going to transcribe genes to crank out all the proteins that we need for cell cycle activity. But as soon as we get past that early part of the cell cycle, you break down those cyclins, and so therefore you stop making those proteins. So we just make enough to get through the cell cycle, right? And then the system kind of shuts itself down, right? So the breakdown of those cyclins stops the functionality of the MPFs, the maturation promoting factors. That's the main way that reset the signal. how did photorespiration evolve in plants? I mean, I think it said it more than likely occurred when there was far more carbon dioxide in the atmosphere than oxygen. So there wasn't really enough oxygen to compete with the carbon dioxide, so it wasn't that big of a deal that Rubisco could bind to both oxygen and carbon dioxide. So the problem is now there's so much more oxygen that if you get into a situation where you can't bring in carbon dioxide because you've got the stomata closed off, then you build up an excess amount of oxygen on the inside and you don't get enough carbon dioxide in there. So then photorespiration takes over. Again, it's a great example for to show that evolution is not going towards perfection, right? Because obviously plants would not want that, right? They've had to come up with other ways to avoid photorespiration. That's what the C4 and CAN plants are. You know, they are the result of evolutionary adaptations to deal with photorespiration instead of just getting rid of photorespiration. But we can't get rid of something, right? So if Rubisco can work with both carbon dioxide and oxygen, we can't just say, hey, stop working with oxygen. So that's, you know, the way, as far as we know, you know, no one was there 500 million years ago, 750 million years ago when plants started to colonize the ground, the terrestrial landscape. So who knows exactly why how photorespiration evolved, but that's our best guess. How much do you know about microfilaments and microtubules? I mean, I would, you know, just know that they're both part of the cytoskeleton, but they serve slightly different roles. So microtubules' main role is to give the cell overall its structure, like it's the big girders that hold the building together, right? It is basically going to make sure that the cell doesn't collapse in on itself. But they also serve as the highways, right? So for John the motor protein, if he needs to carry a vesicle or a vacuole from one place to another, he's going to have a microtubule highway to walk along. They're hugely important during cell cycle activity, right? During mitosis, because that mitotic spindle, those spindle fibers are microtubules. They're going to have to be, you know, really important from that standpoint. Whereas microfilaments, you know, they do play a role in the cell cycle. Those actin filaments are going to help make the cleavage furrow during cytokinesis and finish off separation of the two new daughter cells into their own individual cells. They also make up that the portion of this just underneath the cell membrane of the cytoskeleton that maintains the shape of the cell. And if the cell needs to change shape, if it needs to move, then you're going to have to rearrange those microfilaments, those actin filaments there. They're also hugely important in muscle structure in larger organisms. How does allosteric regulation work? Well, so allosteric regulation, break it down, right? Allosteric means somewhere other than the active site. So for allosteric activation, it simply means that I'm going to like plug something into my allosteric site, and that's going to lock my enzyme into its most active form. So I don't think I gave this example during first period, but I know I did later on in the day. If you think about the front door in the classroom, right, as being the active site, and the enzyme activity is facilitating the movement of people through that door. If you come to the door, you have to grab the handle and open it up. That's work that you have to do to get out, right? But if I put a doorstop in and I make sure that that door stays open, that is its most active form, right? So a doorstop, I shove that in there, that's basically locking the enzyme into its most active form, where it's most likely to do the job it's supposed to do. So allosteric stimulation or allosteric activation is like that. We just take a small molecule and we basically stick it into a site on the enzyme other than the active site. But where I do that, it locks that enzyme into its most active form. So it's going to make it more likely to be able to bind to its substrate molecule and facilitate the chemical reaction, speed up that chemical reaction. For R groups is the only thing we need to know which is polar and which is non-polar. Yeah, I mean, again, if it's got a positive charge on it, then it's going to be basic. If it's got a negative charge on it, then it's going to be acidic, and those can form those weak hydrogen bond. I mean, weak ionic interactions. But yeah, for the most part, just understanding the difference between the polar and the non-polar is important. Mostly, and again, it's not mostly identification, right? I talked to you guys about making sure you understand in those structure and function questions that it's, if I asked you to describe the structure of an amino acid, one thing, or of an enzyme, one thing I would want you to do would be to talk about the amino acids in the active site. That is the most important part of an enzyme, is the active site. Right? And so from a structural standpoint, the structure of any protein, it is at its basic level, it's those amino acids. So if I want to know about the structure of a protein, you need to talk about the amino acids, and the part that gives the amino acids their most chemical reactivity is the R group. So it's more of a being able to talk about it in a way where you would be talking like kind of abstractly about the structure and function of an enzyme. I'm not going to ask you to identify the different amino acids. Justin, you don't need to know the exact inputs and outputs of glycolysis and Krebs cycle, so don't go back and try and memorize the numbers of all the ATPs and all the NADHs and FADH2s, that kind of stuff. Mostly just generically what's going on. We're removing electrons off of larger molecules to try and put them onto those electron shuttles so that we can establish that electrochemical gradient. Well, I hope everyone enjoyed this. I hope it was helpful for everyone. And if it was, then let the folks at LearnLink know. Let Dhruv and Akshay know, and we will maybe try and set some more up for second semester and see if we can to do more of this to help you guys out. I'm glad you enjoyed seeing Chester.
Yeah, so Mr. Sharp, I mentioned to you that this call, it was recorded, but would you like for us to post this to YouTube? If you don't want us to, we won't. But it's up to you. If you do not want us to, we won't. But if you want to, we can.
That's totally fine. No, I told all the kids that if they couldn't make it at 5:30, that you guys were going to be recording it and posting it, and totally fine with me.
Yeah, so I'll send it to you, and you can play in Google Classroom within the next hour. So.
All right, sounds good.
I really appreciate for everyone coming out tonight. Thank you.
Yeah, everyone, make sure you say thanks to Millen and thanks to Dhruv and Akshay for putting this together.